Hi from Berlin. My name is Matthias Rillig, and today I want to talk to you about global change and soils. Now, one question that I've been asked a few times is, what global change factor is worrying you the most? Well, what would you answer? In the next 20 minutes, I'm going to give you my answer, which is that it's not any one factor of global change that worries me the most, but just the combination of many factors acting concurrently. So let's talk about the impact of global change on soil functions and microbial diversity. First of all, it's important to realize why soils. Well, soils are a major target of drivers of global change. In many cases, just by virtue of gravity, everything falls down. But also soils can be part of the solution, for example, by storing carbon, but they can also potentially make things worse by releasing CO2 when not managed properly. So many reasons to think about soils and global change. So what's the issue with many factors of global change? Well, if you were asked about global change in soil, you would probably think about the famous four, like most people. You think of warming, nitrogen deposition, drought, elevated atmospheric carbon dioxide. There was a very good paper in 2017 by Bernhard et al. Who made a very good argument that chemical pollutants or many synthetic organic chemicals should be included in this list of global change factors. So we add a lot of synthetic organic chemicals. And you've probably heard about plastic pollution being an issue, especially because of the creation of microplastic and nanoplastic, something that our lab has worked on a lot during the last years. Shown here is a soil aggregate from which you have sticking out microplastic fibers. So then you add microplastic to the list. But really it doesn't stop there. You can add salinity, soil compaction, heavy metals, artificial light at night, invasive species, and so on and so on. There's a lot of factors when you think about it, and potentially many or all of them can be acting concurrently on certain soils, threatening soil health. So what do we know about all these factors affecting soils? How well specifically do we understand multiple factors and their effects? So we looked at thousands of papers, screening them and looking for experimental work that looked at soils and examined factors of global change. And we counted how many factors were used in each experiment. And the result was, as you might expect, the vast majority of papers used just one factor. A lot fewer used two factors, and almost none used three or four. And this trend was basically pretty stable through time, didn't change at all. And when we looked at it in more detail, we found out that also certain factors are preferentially combined with other factors. And here we have our famous big four: temperature, water, carbon dioxide, and fertilization. They are preferentially combined with each other. And it just drives this point home, 98.2% of papers describing experiments in soil in global change included only one or two factors, which means we know very little about what happens when we have more factors. So why is this work not being done? I can think of four reasons. First of all, there's logistic constraints related to gearing up to studying some of these factors. Just imagine CO2 exposure problems or systems and invasive plants. Many people will just also rely on the thought that, well, by capturing the effects of the big four, we've basically got our bases mostly covered. There is research compartmentalization because global change has become such a huge field that people that work on, let's say, invasive species would not look at toxic chemicals. And most fundamentally, there's the combinatorial explosion problem, which states that with an increasing number of items, the number of combinations of these items increases very, very rapidly. So imagine you had 10 factors to examine, and you had only two levels of each, so on and off, or low and high. Well, then you would have 2 to the power 10 treatment combinations. That's 1024 different combinations of treatments. And then you need to replicate, so you can see there's simply no way we can do experiments like this. And this is a major problem why we know so little about such many factors effects. So what did we do and what did we find? Well, we did this experiment using small microcosms that contain about 30 grams of soil, and we exposed them to a wide range of global change drivers. And they are shown here. We had a control, we had drought, we had end deposition, temperature, microplastic, glyphosate, antibiotics, fungicide, copper, salinity, and insecticide. And here just the results shown for water-stable aggregates and ecosystem process rate. As expected, some of the factors have positive, negative, or neutral effects. So, but what did we do to look at the combinations of factors? As we just explained, we simply cannot do the factorial combinations. There's too many factors to combine. So what we did instead, we had a pool of 10 global change factors, and we randomly drew from this pool of 10 global change factors to include in each replicate. So for example, for replicates of the factor level 2, we did repeated random draws from that pool of 10 factors and included randomly drawn factors in these replicates. So for example, one replicate of factor level 2 would have copper and fungicide, the other one would have warming and salinity, and so on and so forth. So what they would have in common is that they are 2, but the composition would be completely different. And so when we did this in the experiment, this is the result in the shaded part here. This was quite stunning, because rather than being complete chaos, keeping in mind that really what we're doing here is comparing apples and oranges within each treatment, right? We're comparing completely different things. And there was actually not chaos at all, but it was a very, very ordered decrease in water-stable aggregates, for example. So there was a very clear progression of that treatment effect. And analyzing this statistically, we basically could find out that additive, multiplicative, and dominative null models could not capture these responses really. And that just by knowing the number of factors, this was done using machine learning, we could explain about 50% of the variability in the experimental results, and then more when we took into account factor identity and effect size. But there were also some surprises. For example, soil water repellency, which is simply the time it takes for a droplet of water to infiltrate into a soil. There was basically no effect except for a little effect of drought, which is well known in these sandy soils, on water drop penetration time. So basically no effect. But then look at when we had more than 5 factors, this parameter shot up, basically out of nowhere. We didn't see that one coming at all. And notice that this effect only really significantly materialized with having more than 5 factors included. This is well outside the range of what people do experiments on, as we have just seen in previous slides. And here our ability to explain the effects of the many factors from the single factor effects, of course, is super low, because this came really out of nowhere and it's a complete surprise. And we had similar effects on other parameters. I showed you aggregates on the top and water repellency here, but we also looked at decomposition rate and respiration rate. And in every case, there was this ordered response that could be predicted quite well by just knowing the number of factors. And also, a similar picture emerged for fungal biodiversity. So we sequenced the fungal community in there, and for amplicon sequent variant richness, you can think of it in terms of species richness, there was also a steady decline in species number. There was a steady trend in terms of composition of the fungal community, and there was a pattern equally well resolved also for dispersion, so the dissimilarity of communities within each of the treatment levels. And actually the losses here in diversity were mostly due to Basidiomycota taxa being lost from our community. So it was not random, it was specifically affecting certain subsets of the fungal community. So what could this really be due to? We don't really know, because that is one of the drawbacks of this design. We cannot really pinpoint this to any particular factor interaction, right? But we saw that with an increasing number of these drivers or stressors, there were decreases in ecosystem process rates and also diversity. And what could this be caused by? Well, when you have more and more and more of these factors included, these factors could interact directly with each other. You could have a selection effect, which we call the increased probability with more factors to have one factor included with a disproportionately strong effect. Or you could have complementarity effects in terms of multiple factors affecting the same biodiversity component or the same component process. And all of these would basically filter down to affect an ecosystem process rate that we measure. Was this a one-off result? No, we've since repeated it with actually a lot more experimental units with slightly different factors included, a slightly different design and also a different soil. And we essentially find the same response, and also including as an additional parameter high and low soil biodiversity. This work done by Dr. Gao and Yang in the lab. And we're currently testing the same idea also for multiple chemical stressors with a slightly different experimental system that's depicted here. So how can we make sense of all of these factors? So what approaches do we generally use in biology when we have to deal with a large diversity of items, such as the ones here? Well, you look at the different items, you might pick out, well, some of them are animals, and well, some of the animals live on land, and so on and so forth. So we go through and we order and classify these items, like we do with species. And so the question I asked myself is this: can we do this also with global change factors? Can we classify and order global change factors and then bring them together in terms of their similarity? And I did this, helped with Matthias Rillig, who did the statistics, and Annika Lehmann, who did the depiction here, was published this year in Global Change Biology. And so the 30 factors, this factor pool of 30 factors here, I classified according to the nature of the factor. Was it biological, physical, or was it chemical? What was the proximate effect direction? So was it positive or negative, and also on what component in an imagined plant soil system? What was very coarsely the mechanism of the effect? How did it act on the system? Was it a resource, for example? And also some key properties, like was it pulse versus press? And actually what's depicted here is, with the different shading, is the actual data that went into this categorization. And the results of this categorization of basically the tree that came out of that, there's a similarity grouping, is shown here on the left panel. This is just the 30 factors grouped by these 30 traits, and you get some factors grouping with other factors. Now this is a very nice for conceptual integration of a large range of very divergent global change factors. Is probably useful already in and itself, but does it actually mean anything in terms of prediction or understanding experimental outcomes? So what we did, this were 30 factors, as I said, 10 of which we had actually included in our design that I just mentioned to you in our experiment. So on the left is basically just the reclassification of these 10 factors that were included in our actual experiment. And on the right-hand side of this right panel here, these are all of these data that I showed you before taken together, the experimental data from the specific experiment. So you have on the left-hand side, you have the very abstract classification based on first principles, not related to any experiment whatsoever, just very coarse. And on the right-hand side, you have the specific results of one particular experiment was done with one particular soil and its biodiversity, and also its strength of factors and all the choices that were made during this experiment. And we basically asked ourselves, is there a correspondence between these two trees, the abstract factor tree and the experimental result tree, that is better than you would expect it by chance alone? And the answer is yes, there is actually very good correspondence between these trees. Not perfect, but I think it's encouraging that there is this similarity, because it suggests that maybe this classification of factors or grouping factors by their similarity may be a very promising way forward to designing future experiments on this particular topic and sort of taming that immense diversity of factors that we have to deal with here. So what does this all mean? Well, what it means, first of all, is that we got our hands full. We've got a lot to do. We know very little about soil responses to these many factors that act concurrently. We didn't, for example, understand why there was this sudden increase in soil water repellency, and we don't really understand what contributes majorly to these responses that we observed. So we need to think a lot more about this concurrent action of factors collectively as a community, because there may also be just some fairly ugly surprises like the one that we saw in our experiment that are hidden basically behind these very high level factor interactions. And we definitely do need new ideas and new experimental approaches to tackle this issue, like the random sampling from a pool of factor approach that I introduced you to earlier in this talk. And what can we do to help? Well, one way to look at this was that with increasing number of factors, there was an increasing loss of ecosystem process rates, functionality, and biodiversity. But another way to look at it is to look from the other side of that curve. It's like the more factors we can basically exclude from the system or switch off, the higher the chance that that system moves again towards higher functionality and biodiversity. So basically the message is, and it's not borne out by empirical data, but just as an idea, if we reduce the number of factors acting on our soils, this will probably help. And also this sends the very positive message that every measure probably helps, that you can switch off some of these factors that our soils basically have to deal with. And maybe we can use some of the same design, this random combination of maybe management factors or recovery or resilience or restoration factors to move that system back towards higher functionality and higher biodiversity. And we thought about this first in a paper in New Phytologist from a few years ago, where we looked at the total number of possible treatment combinations on an agricultural field, which is likely in the many hundred thousands. And by looking at some of those interactions of factors, maybe we can move some of the systems back towards a more positive trajectory. And with that, I'd like to thank you for your attention. The picture here is our lab. These funding agencies supported some of the efforts that are shown here today. And I'd like to especially thank Annika Lehmann for the visualization, Masahiro Rio for some of the statistics, Julien Roy for the molecular data, and all these other people also made significant contributions. So I believe these multiple factors are really what put soils at risk globally. But I also believe that these many factors will not manifest themselves everywhere to the same degree. I mean, the design of the experiment I talked to you about was mostly inspired by having in mind a agricultural soil or a soil from an urban area. So there will be also a lot of spatial variability in the degree to which factor dimensionality is really manifested out there in real ecosystems. But I think the problem is real, and it's definitely worth our attention. I also don't think we should stop working on individual factors. We need our mechanistic understanding of these individual factors to build on that, to understand multiple factor effects. But I think we need to just include also some component of these multi-factor effects, and I hope that this talk convinced you that this is an important thing to do. So I look forward to your comments and the discussion.